Hi, I'm Benita.
Hi, I'm Shumon.
And today with us, we have Dr. Alfanso Martinez. Alfonso is a developmental biologist. Over the last 15 years, his work has focused on using embryonic stem cells to explore cell fate decision during mammalian development. Recently, he and his team have developed a novel experiment system, which is changing the way to study early mammalian development. From 2003 until 2021, he has been a professor of developmental mechanics in Department of Genetics of the University of Cambridge in UK, working on developmental biology with fruit flies, Drosophila, and more recently on embryonic stem cells.
His interests covered the interface of cell and developmental biology with physics and engineering. He is a member of EMBO, has been awarded 2 ERC Advanced Investigator grants, and in 2012 received the Waddington Medal of BSDB for his contributions to British developmental biology. We are very excited to have you with us today.
Hello, Suman and Benita. Thank you very much for having me here. I'm very intrigued about what's going to happen here in this dialogue. But more importantly, I'm looking forward to having a good time talking not so much about myself, but about science and about what young people like you guys are interested in, and how people like me can help you develop your interests and your dreams, because in a way that's what science is about. It's about making the dreams come true.
So our first question is that how did you fall in love with science? Was there a particular incident that reminds you of the moment?
Well, that actually what you're asking me is a little bit about my trip from really not knowing what to do in life to what I'm doing now. I don't know that there is a moment that one falls in love with science. I think it's part of a continuum. One day you realize that that's what you want to do, that's what you are doing. I grew up in Spain in the 1960s, in a family Spain was a very difficult place to live at the time. We lived under a dictatorship, and also I came from a middle-class family that was nothing to do with science. They were journalists. I was the oldest of 4 brothers. We went to school, and in those days, particularly in that Spain, teaching, learning was not a very exciting enterprise. We had to learn things by rote. It was all very directed to just passing exams, getting a degree, finding a girl in my case, getting married, settling down, finding a job. That's all people develop from you. But the fact is that one had a lot of time. I did my college in humanities, largely because my family had to do with humanities, and I studied several years of Latin and Greek, but slowly, I think I didn't feel that I was good for a science. I told you that we were not nurtured in that way, and I didn't come from a family of scientists. But then I started being curious about science. I felt that I could read books and evaluate them about literature and whatever, and that there was other things in life that require a little bit more exercise. So when I came to university, I decided to switch and to study biology. And that was a very, very important switch. And still, those years were very, very difficult. That was the early 70s. There was a lot of change in Spain. And during my first 3-4 years in university, we hardly had any lectures because the police was always running around the university. It was all but of course, that gave me a chance to search for things myself. I always tell students when I'm with them whether they are bored. And I think the story that you had told me about yourselves and about the kitchen, that's very important, because only when you are bored, you actually find out what you want to do. I think one of the problems with today's world is that there is so many targets, there is so many objectives, there is so much. The time is very busy. You never have time to think and ask yourself questions. So I think it was in that way that I started reading, and I always found funny that we were forbidden to read the books of certain authors, but there was a book that was very important to me that was Chance and Necessity by Jacques Monod. And that was a very remarkable book at the time. And that completely changed what I wanted to do. I discovered Spain at the time also, biology was very classical in terms of plants and animals and all these things. But all of a sudden I discovered these molecules. And the way Monod talked about them was very interesting because he was mixing a little bit of philosophy with a little bit of biochemistry and with very revolutionary ideas. He was a communist. And that was funny, you know, in a country that was very busy, not allowing us to think about communism. A lot of books about communism were being suppressed, but this one was allowed. That was a very, very important book for me. And I became very, very interested in biochemistry, actually. Also, as I said, biology was very, very classical. But in the last 2 years of university, you could choose other things that you would study. And I decided to go to chemistry, and I studied physical chemistry. And I became extremely interested in the interface between physics and biology. That was very, very important. In my upbringing, my father, who was a journalist, always said that I should leave Spain, that Spain was not a country with any future. And he almost programmed me to leave Spain. So when I finished the university, I applied to a Fulbright fellowship. And on the second time of asking, I got the Fulbright and I went to America to do my PhD, which, like you, Benita, you told me before, in Spain, people didn't know what were PhDs. They didn't know that you could do this thing. But I had particularly a teacher in the university who knew about these things and guided me. And he is the one that suggested the Fulbright and the one that suggested to me that if I was interested in this physical chemistry, physics, and biology, I should go to the University of Chicago, which is where I went to do my PhD. And there it was a big culture crash at many levels. I think today it's very difficult to explain to some of you. No iPhone, no Zoom, no nothing. You took a plane, you landed in a different era, because that's what it was, from Franco's Spain to Chicago. And there you were trying to, and the first 2 years were very difficult because all the ideas that I had, I went with a very idealistic view of bridging gaps between physics and biology, but it was very, very early. By the time I also had already decided that the big problem of biology was development, and that I would like to solve it by bringing together physics and biology. But after 2 years there, everything was a disaster. I think that the late 70s, it was very, very early days to do this. We didn't know even what genes were. I mean, we didn't know that development had anything to do with genes. So I did my PhD on molecular biology, which was at the time of enhancers in yeast, and I learned. And then when the time came to do a postdoc, I decided to really do developmental biology. By then, I had heard of one experiment that completely took over my mind, which is the experiments of Hans Driesch. And you may recall that what Driesch did was to take an embryo when it was 2 cells, separated the 2 cells. And instead of 2 half embryos, what he saw is 2 little embryos. And he was absolutely bewildered by this. And that completely dumbfounded me. And I learned, and I said, well, this is really a big secret. This is something very wonderful, and I'd like to do it. So when it came to do a postdoc, I got married, and me and my wife wanted to live in England. And I was very lucky to get the advice to go and work with Peter Lawrence. Now, this was a very exciting time in developmental biology. And there is one thing for everybody, for all the young people, and for you only realize how exciting the time was when you look back into it. when you are in the middle of it, it's all a lot of hard work, it's a lot of expectation. But when you look back, you realize, wow, that was really a very important time-changing moment, because that's the time that all of a sudden Nusslein, Vollhardt, and Bischoff had produced their screen of mutants. People were starting to clone genes. We were starting to get a glimpse of development. So I started to work in Drosophila. That was in 1983. And things went well. And I was incredibly lucky. I think I've been incredibly fortunate to be able to spend almost 40 years in Cambridge and really enjoy the environment there, which is wonderful. And I started to work with Drosophila, and it was a very rich time. And during that time, we learned a lot about genes and development through the fly, as many of the people that are listening know. And I enjoyed being part of it and making some contributions. But then at the turn of that question of Driesch, that experiment of Driesch always amazed me. And then I learned about chimeras. which is that you take embryonic stem cells that they are on a plate doing their thing, you put them in a mouse and they make a mouse, but you cannot get them to make a mouse when they are in the plate.
Yeah.
So I was interested about this, and you could say, well, why not in Drosophila? Well, Drosophila, you cannot do that experiment with Drosophila for reasons that we can talk later. And I really was curious about how this happened, because either this was magic or we were missing something very important. Okay.
Yeah.
At the time also, biology became quantitative.
Yeah.
It started to be very quantitative, and people, so
Yes.
I started thinking that I needed a system where I could be quantitative in a manner that would allow me to understand these experiments of Drish and the experiments of the chimeras. And that's why I turned into ESLs.
The experiment of Trish, actually, I mean, you reminded me of that, and that kind of gave me goosebumps right now, because I was equally fascinated when I read it in actually Dr. Scott's developmental biology book.
Biology
Yeah,
book.
and it was so amazing. Yeah.
It was so amazing.
That.
It was such an amazing experiment that Drish could not find an explanation and became a vitalist and became a philosopher. In fact, I recommend everybody to read a passage in his, there is a book in English, the Science and Philosophy of the Organism, where he reasons. It's a fantastic piece of history, because he is confronted with this amazing observation, and he starts thinking, what kind of a machine could do this? And when you read it, he's describing a modern cell. But in the 1901, those things could not exist. So beautiful. And he says, this thing cannot exist.
It is usually very difficult to manage work with life, and we often struggle through that. Like everyone, no matter which field we are in, we are struggling with it. And how do you find balance between your work and life?
That is an interesting question. And I think that perhaps the simplest answer to that question is that what scientists do is not work. I mean, I think I do understand that sometimes some people need to find a balance between work and life. And we might come back to some issues associated with this statement that I'm doing, because I think something is happening in the science, which is what is creating the problem. But I've never looked at my work as people would see what we do as a job, as something that is work. So, I mean, it's a continuum. I mean, my wife often says, you work too much. No, I never worked, actually. I've never done work. What I do is to think about the questions that I'm interested in, and I weave that in my life, in the normal life. And I have a life in which there is this background of my thoughts, of my lab, of my science. There is no need to balance. I mean, the best balance, and I've been very lucky that I have a family, that we are together, and that provides the perfect combination. In a way, I have two families. One is the lab family, and the other is my real family. And I think it's to balance those two is perhaps a bit more difficult than to balance something that is not work. I mean, I think any scientist that you ask this question to, they will tell you that what do I have in my mind all the time? Well, what are those cells doing? What's that gene doing? Why things didn't work today? But that's in the background, and it's a seamless thing. So I don't have to balance that. I think sometimes I have to curb to hold back the passion, because there are things that sometimes are more important than answering those questions. I think a big problem today is that science has become a job. And I think there are reasons why that has happened historically. And I think that it is a pity, because many people approach it as I need a job. This is what I've been trained for. Therefore, it is a job. I mean, the other day, somebody on Twitter asked a question about what part of a PhD is the training part. Somebody was asking, teaching classes is not training, writing a dissertation is not training, doing research is not training. This person had missed completely the point that science is not a job, and that every one of those things implies a training. It is that sometimes the training, it's what we call mentorship, comes from abroad. But any of us old guys have had to learn these things from ourselves. And I think, Benita, by what you've said, you know what I mean by that. The problem is that today, the educational system streamlines people into this. The PhD programs are just a set of boxes that you have to tick. It's become very, very difficult. So I'm not surprised that it is. But I mean, so I did say, and I don't mind, what is then science? Well, the science is a passion. Science is not a job. Science is something that makes you wake up in the morning and want to the lab to see what's going on, or to open your computer and see what's going on, and keeps you awake at night, sometimes later than you should. That's what science is. If you don't have that and what you want is a job, there is many jobs, many of them related to science. This is something that has happened today. That there is a wealth of jobs that research might not be for everyone. But I think it is. I think that's why you probably get this difference, because the young people have been educated with a set of expectations that I don't think corresponds to science.
The next thing I would want to ask you so failure can be tough. I hear a lot of people telling me that it's okay, you have to deal with failures, but when it comes, it's really difficult to deal with. So how did you deal with your failures?
That's a good question. And it's not an easy one to answer. Anybody that says that they haven't had failures is that they're not being honest with themselves. I think sometimes failures happen because circumstances drop you into a very difficult situation, which is not what you expected. And that has happened to me 2 or 3 times in my life. Other times failure happens because you are too hard on yourself. And you're not living up to your expectations. Those are easy to deal with. A good friend can always tell you that maybe you're being too hard on yourself. But the first ones, the ones that are out of your control, those are more difficult, you know. And I've been 2 or 3 times in those situations at the risk of losing the job or losing funding, which we said that science is not a job, and I just mentioned a job. I mean, it's a curious job in the sense, but those are very, very difficult moments because you want to give up. Maybe you made a mistake. I mean, I remember I had a very small lab. Those were the days where we cloned genes. You had to clone a gene, and I chose one gene in the beginning, and I had a very small lab. And after 2 years of chasing this gene, there was a small genetic error, and I was long way away from the gene. And then what do you do? In those days, everybody cloned genes. And if you didn't have a gene, maybe this is my problem with genes sometimes, that they were never kind to me, and I was never. But I think that what do you do then? You have to be very strong. You have to remember what you are there for. You have to persevere and then trust your luck. And I'm honest there. I feel I've been very, very, very lucky, and hope that people can help you out of that hole. But that's inevitable. I think this is another thing in the context of what you were saying that we are finding today very difficult, because there is so much to do. You have a lot of techniques. You can do a lot of things. It's very exciting. In those days, you know, it's in a way like I've supervised 2 students as a collaboration with crystallography, and with crystallography is very funny. AlphaFold have put out of a job to everybody, but in the classical crystallography is very funny. I don't know how much you know about that, but either the protein makes a crystal or it doesn't make a crystal. There is very little that you can do, and people get very frustrated, you know. So I think you just have to be very, very. It's very, very difficult because, as you've said, as you two have said, because it is our passion. So all of a sudden to feel that because of an element of chance, they're going to remove from you the opportunity of doing it, it's very hard. I mean, I think it's easier when you are fired from a job, you're worried about the security, but when they're about to take away the possibility of following your passion, that's very scary. I think all you have to do is be patient and hope. And I think in my case, I've been lucky.
Continuing from the failures that Benita just asked about, that you had certain phases where it was very challenging. Would you like to talk about those circumstances which were the most challenging phase of your career?
I think there have been 2. Maybe I can talk about one. We'll see if you want to talk about the other. But actually, the first one came after 2 years into my PhD, because as I said, I went to America with a lot of great ideas about how to bring together physics and biology, and that's something that then I did in Cambridge later. And We can talk about how that evolved. But anyway, to go back, so in America, I arrived with these great ideas, but after 2 years, it all collapsed. I met also a very aggressive and somewhat difficult supervisor, which made my life very, very difficult in one of the rotations. At that time, you had to make rotations. I only had my funding was from 1 year to another. I was enrolled in a graduate program, but my funding was not secure. And I was doing a rotation in his lab, and he had told me that I was going to get the PhD there, that everything was fine. And one day he called me in the morning. He had called my committee and told them that I was totally useless, that I should go back to Spain, and he would remove all my funding. And there I was, 2 years into my PhD in Chicago without nothing. I mean, it was very, very difficult. I thought of folding everything, going back to Spain. And a good friend convinced me that wouldn't be the case, that I should look for another supervisor. My head of department was very helpful. They found money for me, and that changed my life completely. But I should say that was very, very difficult because of what we were talking about. I had a dream. Looking back, I realized that I probably had made some mistakes because of self-teaching. I think you know about that. In Spain, I did build this world which didn't exist, and it all came collapse on me. I don't know why this person didn't like me, but I'm glad that with the help of the head of department and a good friend, I managed to pull through and finish my PhD, do a PhD, finish a PhD, and being able to be here today with you. There was one other time where I was also about to lose, in that case, the job, because this is something that today is even harder than it was at the time. In the UK, you have a lot of people on fellowships that they keep on being renewed or not renewed. And there is too many people in those fellowships, because at the end of those fellowships, either you get a job, now we are talking in the jargon of today, or you have nothing. And so I was in one of those situations for a year, but I'm very glad that it went through. And so those were very similar times. Is what we said before, is the fear of all of a sudden losing the opportunity to do what we like and what we love.
Thank you for sharing that experience with us.
Also, I think it gives us so much hope, because I think being an international student, I hear a lot of people say that, the first instinct after failing is pack your bags and go home. But I found very good people in CCMB who actually appreciated my excitement.
Absolutely, and that's probably what you are. And I think this is what is very nice, is to have the opportunity of expressing what you are. That's what science is. Yeah. I don't think there is a way. I mean, one of the things that bothers me a little bit today of all these fellowships and all these interviews and all these schemes is that I remember one day, it was actually the contrast, I don't mind to mention them, that they put out, if you want to do well in our interviews, here are a whole bunch of tips. What you're doing with that is eliminating anybody that it's interesting. Because you are making everybody the same. And I think this is one of the problems with science today, that the people that should be doing a science sometimes are not in a science. And there is a lot of people that should be doing a science that are doing a science, but there is a lot of people that are just ticking boxes in those interviews.
So coming now to the science part in your life, that when did you realize that you wanted to work in development, first thing? And second, a part of this question is that a lot of talk has been going on right now that how developmental biology as a field is not very shiny, or it's almost over for us. So what do you have to say about that?
Well, there are two different things that you've said there. I mean, one is, how did I come to be interested in development? I mean, that has to probably, again, have to do with my upbringing in Spain and in school. We had I guess they were called biology lessons, and we had a teacher, and I was always very curious that every time we came to reproduction, the teacher of biology sent us to the teacher of religion, and the teacher of religion sent us to the teacher of biology. So I thought that probably was very, very interesting. There must have been something. There must be something very interesting when nobody wanted to talk about reproduction and biology. And then I used to see I used to see these pictures of the blastula and cells, and so everything was very, very mysterious. In fact, in the university, things became even more mysterious, and that's why I had a very physicochemical and biochemical slant, because embryology was in the final year. In those days were 5 years, and was taught by a nun. So that was very, very, very strange. That made it even more. I never took that course because I didn't know, but I certainly was interested in the way an egg or a fertilized egg would become an organism. But I thought that we could solve it by bringing physics and chemistry together. I mean, and that's really why I went to Chicago to try to do that. Of course, we are doing that now, but that's 40 years later. I mean, I had this idea years ago, but it was so I had to settle after that crash that I mentioned to you before. I settled to learn gene expression. Then I had my chance to move into development when I went to Cambridge. But I just by then, I think we had to learn about genes. And I spent a long time. Then in about 2000, in a very special conference that I was invited, I heard about the work of Michael Lelovitz. And that was very remarkable to me, to hear about heterogeneities, about noise. And I thought that the answer to Drish experiment might be there. And that's when I decided that we needed a system that the flies did not provide, that would have that plasticity and that ability to change and to be regulative, and also to be quantitative. And the ESLs were that system to me. At the time, the ESLs were starting, and together with a few people, not very many in the UK, we decided that maybe the ESLs would be a good system to actually ask these quantitative questions. And I really want to mention and to be very grateful to Austin Smith, who at the time he had moved to Cambridge from Edinburgh, and he was extremely helpful in teaching us to do ESLs properly. So I think that's the way I switched. I can't remember the second question, Bignita.
Yeah. So the second question is that now there's a lot of conversation going on about how developmental biology is not very shiny, and how the field is like degrading with time. And
so I am gonna be a bit unconventional and honest, because there was this unfortunate article by Claudio Stern, followed by a letter of Peter Lawrence, which I think it was unfortunate and disappointing, because those are figures in the field who should be leading the changes. A science is always changing, okay? A science is always changing. I think this is very important. I like to read history of a science. It's one of my hobbies, and it's very, very interesting and enlightening, because I think you can see where we come from. You can see how the same questions, I think that sentence from Jean Rostand is very good, that the scientist pass and the frog remains, because the questions are there. Maybe we change the angle, but the questions are the same. And I think that we are in one of those changes. So if you think about it, developmental biology is born from the death of embryology. Embryology becomes, which is descriptive, becomes experimental embryology. The great Spemann, Holfreten, Hamburger, Mangold, those are very important. That's experimental embryology. And then molecular biology comes, and experimental embryology runs out of steam. And then molecular biology has to start creating a new ground. And then we get developmental biology, which is a mixture of genetics, experimental embryology, and molecular biology. But, you know, it comes to a point where things are moving now. We have much more powerful technologies, not only at the chemical level, all these single cell analysis. We also have computational techniques. We can ask the questions at a totally different level. We don't need to continue to iterate the whole thing. That requires an openness that unfortunately these people don't have. But I think it's very good. I mean, I don't understand a lot of what's going on in the lab, but I know the questions, and I like to share the questions. I mean, people in the lab know that I feel more comfortable when I don't understand what they are talking about, because then I have something to learn than when they tell me something that I know. As I told the postdoc recently, you know, I know a lot, but half of what I know is wrong. And it is for you to figure out what half that is, because I don't know which one is wrong. I mean, I think it's an amazing time for all of you. And I think it's true that because of the number of people, the number of techniques, the emphasis on publication, which I think is unfortunate, it is very easy to lose sight of the questions. And I think it is those of us who come from a much more limited and restricted time to make sure that those questions are not forgotten. So I think that developmental biology is changing, is morphing into a new incarnation, as it happened before, 2 or 3 times in the 20th century, as I've said before, and now we are moving a much more exciting one. I mean, which your brains are better than mine to grasp it. But I do have something to contribute. Okay, don't forget about it.
So what are the new exciting areas and relatively unexplored areas in developmental biology according to you?
One of the very exciting things that need to be addressed today, I think, is the relationship between genes and cells. I think we've gone through a period which has been dominated by genetics. And I've been thinking about this recently, and I have the impression that we have turned what has been a method to find out the elements that play together to build an organism. We have turned out a method into an explanation because we've been successful with genetics to identify elements. We've decided that that's where the answer lies. Actually, when you look at an organism development, what you see is cells. And the cells are actually, in my view, the cells are using the genes to build organisms. And this is the big question. I mean, there is a lot of this very active field at the moment of mechanobiology. I mean, people talk about single cells, and when they talk about single cells, your mind goes very quickly to single cell RNA-seq and UMAPs. But actually, there is something much more exciting happening, which is more difficult. It looks like it's a totally parallel field, which is the analysis of embryos at the level of single cells, how they are moving, how they are activating genes in groups by contacting with each other. You cannot elucidate that from the UMAPs. You have to do a number of very different measurements. I mean, microscopy, mechanical measurements. I think that's a very lively field at the moment, and bringing the two together, that's going to be where understanding is going to happen. It's not going to be easy. It's not going to be easy, but it's going to be very, very important. I always make an analogy with physics. And in physics, people had described the thermodynamics quantities, pressure, volume, temperature. We all know the basic rules of thermodynamics, but they know that there was a material basis to that. And that's where the statistical mechanics comes. They look at the causes of those variables in terms of the particles. And the statistical mechanics is the way of transforming these microscopic variables, which is the velocity and the position of the particles, into these macroscopic quantities that are pressure, volume, and temperature. I think we have phenotypes that are the pressure, volume, and temperature, and we have the genes at the other end, and the cells are the vehicle that transform one into another. And that's really the big challenge, to find the rules, how it happens during gastrulation in a frog, that you get these thousands of cells, and they start this choreography in which they move in very coherent ways. How do they know that? That's not in the genes. So this is one of the things that are very important. The other, to me, it's very, very important, is that we are getting to the threshold of an opportunity to understand human development, early development, and that's very exciting too. That's going to come along slowly, but I think that's the other thing that it's coming along, and that is going to be very exciting.
Coming to our next question, that is where you work right now. What is the coolest thing about gastruloids? And if you had to explain gastruloids, what are they? How would you do that?
Okay, maybe I should tell you how we made gastruloids, and then you will understand what I find cool, as you say, about gastruloids. So as I told you, I was interested in DRISH, and I was interested in the mosaics, that you put the ES cells in an embryo, and I said, well, can we build an embryo? So I was interested in building an embryo. And the way you build an embryo, a mammalian embryo, is because you can assemble, you can put together the extraembryonic tissues and the embryonic cells. And we tried that, and it just didn't work. We were not very good at that. In fact, other people have succeeded, and I will come back to that in a minute. But then we decided to see what the ES cells would do on their own. And using protocols that we had learned, as I told you, we, together with a handful of other people in the UK, we had started to use the ES cells as a system to study development and developmental decisions. And we realized a number of things that allow us to transfer protocols that were in 2 dimensions to 3 dimensions. And we were very surprised when a postdoc in the lab, David Turner, working with an Erasmus student from Holland, Suzanne van den Brink. David and I had thought of the protocols, and Suzanne was doing experiments, and one day she came and showed us these strange structures that were very, very remarkable because they were organized. And we were just very surprised. I thought that there was nothing inside them, and we started to look at them, and we saw that they were organized in terms of gene expression. But I didn't understand it. They were teasing me. They wanted to make an embryo, but they were making something very, very strange. You were asking me what is that I find most exciting about the gastruloids, that we don't understand them. Although lately I think we are making progress into that they are trying to make an embryo. And I'll come back to that, but they don't quite make it. But then we learn a couple of things. Then I think, and this is very, very important, this moment, that I thought I needed some help. And I think this is something that it's the way I do a science. I see it as a collaborative thing and instead of a competitive thing. And I went to Switzerland to visit Matthias Lutolf. He's an engineer, a bioengineer, and at the time, very, very good. And I went to visit him. And one day I was there, and Matthias said, "Oh, Denis Duboul wants to see you." And I knew Denis because he's an old timer. We come from a time where there is very, very few people. There is, I think, and I went to see Denis, which is always great fun, as you know, and always interesting. And what are you doing here? And I showed him a movie of one of these gastruloids. And he was very taken aback in his very cool manner. I think Denis is more cool. I'm not cool, but he is. And he said, we should look at the Hox genes. And I remember, he might forget, I was trying to get this email. When I went back to Cambridge, I wrote to him and I said, Denis, don't waste your time. Something very weird. Nothing is going on in here. I'm sure we're going to find a mess. And he got very upset with me and said, because I said, this is a very expensive experiment. At the time, I didn't have a lot of money. And I said, this is what you want to do is he said, I spend the money the way I want to. Let's just have a look. And I'll never forget the day that he called me and he showed me the expression of the Hox genes in the gastruloids, because that was amazing. And that was really when I started to think that this thing that we had discovered, against my better judgment, I didn't think that they were interesting, that there was a lot more in there that there was. So then we just, he has been doing his research. We published something together which revealed how much like an embryo they were. From the beginning, we did notice a couple of very interesting things, like, for example, you get what I like to call a genetic body plan, but there is no morphogenesis. So that all of a sudden is separating the morphogenesis from the gene activity from the cell activity. And this is one of the things that is very important. Over time, I think I've realized that I'm very glad that we didn't make an embryo, because if you make an embryo, you've made an embryo. I mean, you know, we've made something that I think is much more interesting, because it's telling us things about what cells do when they try to make an embryo in a very reproducible manner. So it's telling us some things. I think sometimes people, when you talk to physicists, they say quantum mechanics is not intuitive. You have to do the calculations. We tend to think that biology is intuitive. I think gastruloids, for example, tell us that there is nothing intuitive. That we come like organoids. I think one of the most obvious things about organoids that nobody remarks is that we come in pieces. I think it's very strange that you can get a disembodied brain or a disembodied pancreas or a disembodied. I think that's very remarkable, and nobody remarks over that. The gastruloids also point out another thing that I think we are not very good biologists to explain the things that are obvious, and yet they are very deep at the same time. So gastruloids told me something very interesting. You take cells in a Petri plate, you differentiate them, ES cells, and they will become all the different kinds of mesoderm, but after a few days, there will be many cells of one kind, few of the other, they will be becoming a mess, they will be getting all out of time. You put them in that small number of cells, which is one of the tricks of the gastruloids, and all of a sudden the same cells organize themselves beautifully in a space, measuring a space, getting they're the same cells and the same genes, but obviously they are given something very, very important. That is something very profound if you think about it, and it's telling us that cells can do things with genes that genes cannot do with cells. And to me, that's very, very
Yeah.
intriguing. And I'd like to find out what. So I think gastruloids are telling us something about the basic units of life, the cells, which is going to be very interesting to explore. I mean, I see the embryo as the great creation of the cells. You know, is the codron of the organism. That's what the embryo is, and the cells can create that in that codron. So I think that what I find cool about them is that they challenge us. They present us with questions. They are weird because we are used always to interpret biology. Biology is full of stories in a Rudyard Kipling kind of way. But all of a sudden, you know, going back to Drish, what does Drish do? Drish does an experiment that gives him a surprising result. He separates 2 cells, 4 cells, and he gets 2 or 4 embryos. With a gastruloid, we do an experiment which we ask cells what they can do, and we find something which we didn't expect, and that it's probably at the heart of what is an embryo. Okay. One of the exciting things is that recently I think I finally understand gastruloids. It's only taken me 10 years. Okay. But that would mean that would take another. I should tell Iftach, by the way, because I think he'll be very excited.
I
will.
should inform him.
And I will. And I will. And I will. I do understand gastruloids. Anyway. So I think
so tell
yes,
us.
it is
please,
that we don't
I would
understand.
want to know.
Yeah, it's very simple. I think basically one of the questions about the gastruloid is that they don't have a primitive streak. Primitive streak is this very important structure that the reason why they don't have a primitive streak is because they are the primitive streak.
Wow.
So I find them exciting, you see, because what I want, where do I feel comfortable, where I don't know something?
If you were starting your career now, what would be the top 3 questions you would like to address?
There are many questions that one can ask, but there is very, very few good questions. There is an anecdote about Einstein, which I think will answer your question. And he was talking to a French poet, very famous French poet, that was interested in how a scientist works. And Einstein didn't understand what this guy was telling him. So the guy said all of a sudden, he said, well, Mr. Einstein, I'm sure that you must do something when you're working. And he said, oh, yes, I take walks, Einstein said. And the other guy said, and of course, when you go for a walk, you take a little notebook and a pen. And Einstein was very confused by this thing that the other guy was telling him. He said, why would I take a notebook and a pen? And the other guy said, well, Mr. Einstein, because when you have an idea, when you have a question, you write it down. And Einstein said, oh, I understand. No worries. An idea is such a rare thing that if I had one, I would remember it. So I think it is the same with good questions. I think today I think the questions are questions that have remained there for a long time. I think today what I would recommend people is to find what are their questions, what moves them about the real world. But what I would tell everybody to get very, very good at computational techniques and at maths and statistics and computing science. That is absolutely crucial today. Think about the questions, but also get those tools very well under the thing. I think it is a problem that in many biological courses, this is not so important. And biology is being taken over by computer scientists and physicists. I mean, there is today a lot of people that are coming under the umbrella, but if you were starting, that's what I would do. It surprises me that's really what I wish I was young then, because as I told you before, when I went to America in the late 70s, this is the world I wanted to go into. So I think the questions is something, as I said, how the behavior of individual cells that seems to be random leads to these very precise, reproducible, and beautiful patterns. I think that's a very big question, but you're not going to have that with intuition and genetics. You have to develop this computational state. And what is important, the interesting questions, the problems are there. Problems are there. Questions is the way to access those problems. And I think what people, I hope that you guys can tell me what are those methods and those things, because I really haven't found them yet, okay?
So coming to our next question, what do you like to do when you're taking a break from science?
But I never take a break from science.
I for some reason, I just knew this answer. Like,
you know,
while
my head is always going around many things. It doesn't have to be exactly the experiment, you know.
So,
but yeah, I mean,
so
I like to read. I like to read. I like to read. Particularly at the end of the day, I like fiction. It's very important not to be, but I like to read a lot. History of science, I like very much. I like to help in the kitchen with cooking if necessary. I think that's entertaining because I get to drink a lot when I cook. I think that's very good, and to eat a lot. I like football in particular. I can't play anymore, but I like to watch football and be with my family. I think that's very, very important. Although sometimes, if I'm watching a movie with my wife, all of a sudden I have to say to her, what's going on? Because the last 5 minutes, all of a sudden I had another idea about the lab or whatever, and I get distracted. Because as we say, this is running in the background all the time, right?
What are your views on the impact of social media in today's science?
I think it's very positive. I think it's very, very positive. I think the Twitter science community, it's a very good community. I think we should be allowed to be a little bit more critical without being aggressive. I think it's a very good place to discuss things openly, but I think without being aggressive, I think it's all very nice. I've seen trends over the last few years. I find those threads of papers extremely useful. They save you reading a whole paper. Sometimes you have a curiosity and you don't have the time. And I think it's very interesting. I really encourage young people in the lab to get into Twitter. And I try to teach them what I've learned from how to do Twitter, because of course it's not, I'm not saying that one does it well or badly, but I think it's very important to avoid mistakes in the way one deals with people and the thing one says, but it's a very good thing. I think I've got to know people that I've never met personally, but we have good discussions, and sometimes we go in private. It's a very good place to exchange ideas. I find it part of the open science thing, and I think sometimes the thing that overwhelms me a little bit is that sometimes there is too much. I mean, I think you were talking about everything cannot be the best discovery in the world. Everything cannot be the greatest finding. I mean, there is so many histone modifications that I can deal with at the end of the day, and there is so many I cannot. So that also makes you think, if instead of being overwhelmed, you step back and you ask the question about whether there is something here that is worth it. It cannot be all these things. So it makes you think. It makes me think about what are the important things. I find also some of the science journal is very good. I think it's very important in changing the atmosphere in the science. I think it's an instrument of change if we all help. I've seen, for example, bioarchive has become a very important tool of our trade, and it's been. I think social media has been very, very important in that. So I have, in general, a very, very positive, because people, of course, have people in the more pop culture and politics. They have very different experience, but I think the science Twitter community, it's a very, very positive one, and I hope that it will continue to grow.
So since you have mentored a lot of students and you yourself got mentored by people, what is your mantra for mentorship?
There are two things there. I forgot to say something before that is quite important for people, and I'll mention before answering your question. When somebody goes to do a PhD or an internship, you have your supervisor. But I think slowly you need to find another person, which usually it happens, which is your mentor. Sometimes your mentor is your supervisor, but sometimes it's not. I mean, it happened to me in my thesis. There is somebody else who will influence you and who will matter, and someone whom you can talk to in ways that you cannot talk to your supervisor. So the things that you cannot talk with your supervisor. In terms of mentorship, I think true mentorship recognizes talent and nurtures that talent. I think a true mentor does not think of him or herself, but thinks of the other person. I think these are the most important things. I think look at what you have and try to grow it and try to develop it in the way they can. We are all difficult. We are all different. We are all different, and this is very, very important. I mean, I think always I tell the people in the lab that everyone is different. They all have one relationship with me, but I have many relationships in the lab, and each one is special in their own way and requires that treatment. Some people are very independent. They just want your opinion. Others need a lot of care. And if you do that and you bring them together, you'll get more out of science.
Was there a particular advice that was given to you while you were growing and you were seeking mentorship?
Oh, no. my mentor during graduate career, during my PhD, which will be, it's a famous scientist. His name was Jim Shapiro. That was not my supervisor. That was my very, very, very curious character. I think he always challenged me. I'm not sure he gave me any advice, but he challenged me. And it's curious because we remain friends until now. We talk to each other and we see, and that's almost 50 years. And when we were in England, whenever they came to London, we saw him, and that was very good. So I think he challenged me. I don't think, in terms of advice, I always remember my professor of genetics in Spain when I was going to go to America. He said, make sure that you don't invent the typewriter because it has been invented. I think that's very important sometimes. I think
yeah.
What are the challenges of pursuing to be a scientist in the current structure of academia?
In the context of the conversation that we have been having, I think that you should make sure that you ask yourself the question if you are a scientist or not. And for that, you need time. As we've said before, there is very little time. I mean, I've discussed this with my son many times, that they don't have time to ask themselves that question. Sometimes people find themselves doing a PhD without ever asking themselves if that's really what they want. I mean, I sometimes sit with students like you do when you visit labs around lunch, and I like to ask them that question. And I realize many times they haven't asked themselves that question really. And I used to say that in the first year students, when I was in charge of the graduate students, I used to tell them that during this next year, you're going to learn more about yourselves than about the topic that you study.
That is so true.
That's really the thing. I think also, you were talking before about mentorship or supervisors. To me, the students, I give them absolute freedom. I think it's absolutely important because the PhD is the moment where you can play. If you're in a good lab and you have a good supervisor, you'll get a postdoc if you want. When you get to a postdoc, that's more difficult. That's you have to think a little bit more. But as a student, if you have it, you should do it. I mean, sometimes I like to discuss with them whether they can do a science or whether they can do a PhD only. If you can do a PhD, you don't need to do a science to do a PhD. Yeah. But if you do a science, you will get a PhD. And there is all sorts of people. And sometimes people all of a sudden they find there, okay, we'll help them to do a PhD, and then you can decide what you want. But always having fun. To me, that's the important thing. So the advice is to, I mean, as I said, there is students and there is postdocs. I presume that as a postdoc, you've already made the decision. I think as a student, it's a difficult thing. As we discussed before, today is very, very difficult. It's very, very difficult. I think the advice is think very, very carefully about whether this is what you want to do. Today, fortunately, there is a whole spectrum of things that people can do around science. And I'm very excited to see, for example, it's very interesting to tell people that Pixar in Hollywood has a master's in science artistry, in science communication. There is, science communications, a science illustration, podcast. There is a whole. There is also industry. Industry is very, very good. When you think in many labs, after all, what do the scientists do? What do the PhDs or the postdocs? They participate at the very low levels in the projects of the supervisor in the hope that they will get a paper and they will get a job. Well, go and work in industry, but you're going to get a decent salary and you're going to be doing the same thing, and at least you're going to have free time. I mean, there is all this massification of the situation right now. So don't think that a PhD or academic science is the only thing that you can do if you like science. I think that would be for the starters. Ask yourself, what is really that attracts me about this? Then talk to someone or find out. There are many, many ways that you can be, because the important thing, we only live once. And we should make sure that we make the most out of it and not a misery out of it, okay?
What do you think about open science publications?
Well, that's a no-brainer. There is not much to think about. I mean, there is two elements there, right? We need that because the amount of science that is published and the time that it takes to publish something, it's important. I said there is two aspects. One is the ability that preprints have given us to post the science immediately. And I think this is very good for everybody, particularly for students and postdocs, because they see they get their work to have some visibility. So I think it's very good. On the open science, I think if we switch to open access, I have very mixed feelings about that, because many journals are taking advantage of open access to get money in different ways, in two ways. And I think that puts a whole bunch of people at a disadvantage who cannot pay the fees that open access costs. And there are places where the universities or the institutions they take care of that, but there is many places that don't, and many places which restrict that. And I think that restricts that a lot. So I have very, very mixed feelings about the culture of open access, whereas open science, which I would put it differently, I'm all in favor. And I think the bioRxiv has been fantastic in promoting and in developing this culture. And I'm very glad to have been there. Something that many people may not know about bioRxiv is the fact that when you submit a paper to bioRxiv, it doesn't go directly online. There is a group of people, and I happen to be one of them, which have to troll through the papers that have been submitted to weed out those that are not science or that are dubious science. So there is a filter that is very important. So I just think I think we're just making publications. Science has become too much about publications and much less about science. And people obsessed with that, and that adds to the problem of the job, and is mixture with the job. I just wish we could embrace in easy ways bioRxiv. I think it's been done. It's very fast when I look back, and I think at the beginning of bioRxiv, I think it's happened very quickly, but it will happen more. It will happen more. So I think it's the only way that we have. I'm surprised that some people don't trust bioRxiv. After some people, fortunately, there are few.
So there's usually a lot of mysticism among the public about scientists in general.
Of course, when I went to America to do a PhD in science in Spain, science was something very, very odd, very, very weird. It was something for weird people, not something you did. You didn't know what it was. It was a degree if you want, but nothing else. So I think science was an unknown. I think it's become, over the last 20 years, really, companies, genetic engineering, diseases, IVF. So a lot of that people associate with science. And I don't think there is a lot of mysticism anymore associated with science. People know what scientists do. Maybe they don't understand it and they don't appreciate that there is some basic science, although that is complex. But I think in most of the countries of the world today, science is something that people do understand and do see.
Our next question comes to, do you have any advice on how young researchers can bring creativity in their science?
I mean, how can you teach someone to write music like Mozart? Well, you can't, I mean, how can you get someone to do sculptures like Michelangelo or so the creativity is something that you have or you don't have. It bothers me a little bit sometimes when I see online people pontificating in Twitter sometimes about how to do a good presentation or how to do an original paper or how to be creative. Look, guy, I mean, you can have some rules about how not to do boring things, how not to do things wrong, but how to be creative. There is people out there that are very, very creative, and you're never going to be like them. I think creativity, and also something very important in these days, goes back to what we were talking about, the interviews. There is diversity. So I think we have to appreciate diversity. There is no one way of being creative, In fact, the most creative people, when they express themselves, they are not recognized as creative, but as rubbish. It is only in retrospect that they are recognized as creative. So I'm against teaching people to be creative. What you're going to get is bullshit, Sorry.
If you were given the power to change something, what would be that one thing that you would want to change about academia the most?
Well, I think we have a problem. We have a problem, and I don't know what the solution. So if I would change something, is the opportunity to think very seriously about the problem. The problem is very, very, very simple, and it's been sort of a theme in this conversation. I think we are running something very important. We are talking about academic science on a model that was created for science in the last century. It cannot cope with the possibilities, and there are enterprises coming up. There is a lot of people, you heard about these altars that is going to give us immortality, or these other institutes they are creating. So they are being creative in ways an academic sits there in its prehistoric sort of organization and ways, and we keep on thinking that the way to do a science is to do college, do an undergraduate, do a PhD, do a postdoc, write 5 papers, get a job, set up a lab, perpetuate. That is dead. That is dead. It's more dead than developmental biology. I think but the problem is that at least developmental biology is moving, it's being transformed into something else. But this model, all it's doing is accumulating corpses. You know, we are getting all these people that don't have jobs, and some of them are very good, and there is no opportunities. We need to be creative. So what I would like to change is this system that we have. I have some ideas. There are some ideas. One thing that definitely we should encourage is collaboration and admit that there is people, find ways in which labs are not these individual enterprises in which we compete with each other. I mean, the question that I hate in interviews is when they say, who are your competitors? They should ask, who are your collaborators? That's a much more interesting question, because it gives you a more, I think we should create space, and some countries, France has it, where people that don't want to run a lab can still do a science by being put in an umbrella where that happens. So I would like the opportunity to think about how can we change this system so that it can accommodate what we have and we are facing at the moment.
So coming to our next question, what is that one best and worst advice you were ever given?
I think the worst advice, I remember it very well, and it's not, the bottom line of the worst advice, it was a moment to make a decision about where to go. I was embedded in a very nice environment in Cambridge, and there was some people were very interested in me moving out of that environment. And the bottom line of what I was interested, but I had my doubts about leaving behind someone that was a very important mentor as a young PI, and someone that I looked up to a lot. And the bottom line of the advice I was given is be selfish, think of yourself. And I think that's not a good advice. I mean, that's what I remember as the worst advice ever. I think you should. I wasn't gonna sacrifice the potential of doing better for something that I was, an environment that I was enjoying and that I was learning. I think that's also interesting for people when they want to. They are trying to choose an institution. You shouldn't choose an institution because it's the most famous one, or the one that is going to give you a paper, but you should find what suits you, what is going to allow you to grow, what you are learning, what you feel comfortable. You should never betray that.
What would be the one advice that you would like to tell all the young audience who are listening to you?
Enjoy life. You only live once. I mean, you know, make sure, particularly a lot of the people that might be listening to this would be very privileged of having the opportunity to choose. So make sure that you choose not what is going to make you famous, because that is very transient, but what is going to make you happy.
Coming to our last question, what do you think about Kitchen Lab?
It's great. I think it's a wonderful idea. I think you're doing something very important. I think people should listen to the videos and learn from them. And I think you are a wonderful person, and your friend here, Sumit, is really great. And what you are doing is very, very valuable. It's bringing optimism and hope to a world that needs it. We need more things like this, but we need more from you. And that's what I hope that you'll be doing.
Thank you so much. It was so good meeting you.
Thank you.
It was amazing talking to you.